Hello, this is Professor White, and I will be teaching you Mendelian genetics. Let's start get to know first about who Grigore Mendel is. Well, Grigore Mendel is responsible for the laws governing inheritance of traits. Now, he is an Austrian monk, and he studied the inheritance of traits in pea plants. Now, he developed the law of inheritance, and Mendel's work was not recognized until the turn of the 20th century. So this is Grigore Mendel. Now, Grigore Mendel, between 1856 and 1863, he cultivated and tested some 28,000 pea plants. And he found that the plants' offsprings retained traits of the parents. And Grigore Mendel is now referred to as the father of genetics. Now, this is the site of Grigore Mendel's experimental garden in Czech Republic. Now, during his time, he stated that physical traits are inherited as particles, quote unquote. Now, Mendel did not know at the time that the particles were actually chromosomes and DNA that we know of today. Now, here are some genetic terminology that would help us understand this chapter's topic. First off is trait. Now, trait is any characteristic that can be passed from parents to offspring. Heredity is passing of traits from parents to offspring. And genetics is the study of heredity. We're also going to be talking about monohybrid cross, which is a cross involving a single trait, for example, flower color. Or a dihybrid cross, which involves two traits. Let's say, for example, flower color and plant height. Now, we're also going to be learning about the Punnett Square. The Punnett Square is a tool used to help solve genetic problems. Now, as you can see here, there is a bit of a technique in order for you to segregate, so to speak, the gametes from female or the mother's gamete and the father's gamete, or male and female gametes. And this is what we're going to be talking about in a bit. Now, as you can see over here, this is just a quick summary of how to make a Punnett square. So a Punnett square allows geneticists to predict possible genotypes and phenotypes of the offspring. So for this example, parents are both heterozygous. So when you say heterozygous, they have the dominant allele, which is a capital letter, and a recessive allele, which is a lowercase letter. So in order for you to make a Punnett square, you need to make a grid. This is where we can place all the alleles of the gamete from one parent along the top of the grid and those other parent on the left-hand side. As soon as you have segregated them, you're going to combine the parent's allele inside the boxes, and the letters show the genotypes of the offspring, as you can see over here. And we're going to get to know more about this. And in order for you to make sense of it, the law of dominance actually is followed in order for you to determine the phenotypes and the ratio of offspring in this cross in a Punnett square. But we're going to get to that in detail later. Now, what are alleles? I mentioned that earlier, but alleles are the two forms of genes. It can be a dominant or recessive alleles. Now, a dominant is a stronger of two genes expressed in the hybrid, represented by a capital letter. In this example, R, for example. Now, a recessive allele is a gene that shows up less often in a cross, which is represented by a lowercase. Now, when you say genotype, this is a gene combination for a trait. It can be a combination of both dominant trait or allele, or a heterozygous, or it's a combination of the dominant and the recessive allele, or just a recessive allele trait. Now, a phenotype is the physical feature resulting from a genotype. So that's the one that you can see on the outside. Now, here's an example. So a genotype of alleles are capital letter R, a red flower, and the small letter r is a yellow flower. Now, all genes occur in pairs. So two alleles affect the characteristics. So the possible combinations are genotypes can be 2 capital letter R, which is a dominant red, or capital letter R and small letter r, or 2 small letter r. As you can see here, if the genotype is both capital letter R, it turns out to be the phenotype as red. If it is a capital letter R and a small letter r, it's still red. And if it's 2 small letter r, that's when the phenotype becomes yellow. So that's basically what phenotypes are. Now, genotypes can be homozygous genotype. This is a gene combination involving 2 dominant or 2 recessive genes. So in this case, 2 capital letter R's or 2 small letter r's. These are also called pure bred. Now, the second one is your heterozygous. So when you say heterozygous genotype, these are gene combination of 1 dominant and 1 recessive allele. So in this case, it's capital letter R and small letter r. This is also called the hybrid. Now, let's take a look at Mendel's pea plant experiment. So why did he choose peas? Now, peas can be grown in a small area. It produced lots of offsprings and produced pure plants when allowed to self-pollinate several generations, and can be artificially cross-pollinated, which is why he experimented on it. Now, pollens contain sperm and produced by the stamen of the flower. Ovary contains eggs. So the eggs are found inside the flower. And so in this case, it does the self-pollination as we are referring to. So the pollen carries sperm to the eggs for fertilization, and that self-fertilization can occur in the same flower, and that cross-fertilization can occur between flowers. So that's basically what Mendel did in his experiment. So in his methods, Mendel hand-pollinated flowers using a paintbrush, and he could snip the stamen to prevent self-pollination and covered each flower with a cloth bag. And then he traced traits through several generations. So this is basically what he did in his experiment. And then after the self-pollination and all the germination happening, Mendel produced a pure strains by allowing plants to self-pollinate for several generations. And he observed 8 pea plant traits, such as the seed shape, whether it's round or wrinkle, seed color, yellow or green. He also observed pod shape, whether it's smooth or wrinkled, pod color, green or yellow, seed coat, gray or white, flower position, it can be axial or terminal, and the plant height, whether it's tall or short, and last but not least, the flower color, which is purple or white. So these are the different characteristics that Mendel studied in his pea plant experiment. So right here. So this is just some pictures of the seed shape, color, pod shape, the pod color, the flower, the position of the flower, and as well as the length. So in his results, this is what he found. The ratio of dominant to recessive trait in the F2 filial generation is 2.96:1, meaning there's about 5,474 smooth versus 1,850 wrinkled. As you can see over here, you can just read it on your own, what his results were in terms of dominant versus the recessive traits. And the average ratio of all the traits that he noticed is 3:1. So that's basically his study. Now, did the observation ratio match a theoretical ratio? Well, the theoretical or expected ratio of plants producing round or wrinkled seed is around 3:1. Okay, 3 round is to 1 wrinkled. Now, Mendel observed 2.96:1. So the discrepancy is due to statistical error, and the larger the sample, the more nearly the results approximate to the theoretical ratio. Now, earlier I mentioned about generation. So the parental generation, that's basically the generation in a breeding experiment. And the F1 or first filial generation is the first generation of offspring in a breeding experiment. And from breeding individuals from P1 generation, F2 generation is the second generation offspring in a breeding experiment. So this is the second filial generation. And this is from breeding individuals from the F1 generation. So basically, parent generation, F1, you cross those two, and then the F2 is the filial generation crossed together to produce F2. So cross pure plants, and then results in all hybrids, and these hybrid plants are basically crossed to produce these F2 generation and producing 3 tall and 1 short. Now let's now go to monohybrid crosses. So when you say monohybrid crosses, this is basically looking at one trait. So let's take a look at seed shape, whether it's going to be round, capital letter, which is a dominant trait, and small letter r, which is wrinkled. Okay, so we'll be crossing round seeds and wrinkled seeds together. So pure round and pure wrinkled, we're going to cross them. So we're going to segregate these alleles, one on the right, on the left-hand side, and one at the top. And as you, of course, segregate them, then you cross the two, producing capital R and small r on the first box, capital R, small r in the second box. Same here on the next row and the last one. As you can see here, you would see that the genotype are all heterozygous. You also call that your hybrid. Okay, so capital letter R and small r, which the resulting phenotype is round, and the genotypic ratio are all alike, and as well as the phenotypic ratio as well. So this is your P1 monohybrid cross. Now, what are we going to do? The homozygous dominant, the homozygous recessive, all offspring will become heterozygous or hybrids. And the offspring are called F1 generation. Now, all the genotypic and phenotypic ratio is all alike. Now, what are we going to do with that F1 generation? So the trait, which is seed shape, which is, of course, your round seeds, which is already a hybrid, we're going to cross them now into our monohybrid cross. And in this result, as you can see here, there's one window in our Punnett Square with pure, and then heterozygous, and then another heterozygous, and your homozygous recessive trait. Now, how is it different from the first one? Well, the genotypes available for this monohybrid cross is 2 capital letter R, or purebred, and then heterozygous, capital R and small r, and recessive trait, 2 small r. So the phenotype would be round and wrinkled, and the genotypic ratio is 1:1, and the phenotypic ratio is 3:1. Right here. So in this F1 monohybrid cross, we cross 2 heterozygous alleles, and the offspring is resulting into a 25% homozygous dominant, 50% heterozygous, and 25% homozygous recessive trait. Now, what do peas look like? Now, some of these peas have smooth textures, while others are wrinkled, as you can see over here in this example. Now, let's take a look at Mendel's law. Now, the results of monohybrid crosses. Now, inheritable factors or genes are responsible for all heritable characteristics. And phenotypes is based on the genotypes. Now, each trait is based on 2 genes, one from the mother and one from the father. And the true breeding individuals are homozygous. Both alleles are the same. Now, according to the law of dominance, in a cross of parents that are pure, for a contrasting trait, only one form of trait will appear in the next generation. Now, all offspring will be heterozygous and express only the dominant trait. So in this example, you have your capital letter R dominant trait crossed with the recessive trait, all yields to a heterozygous or capital R and small r, which is the round seeds. That's your law of dominance. Now, in this case, this is basically 3 genotypes will yield 2 phenotypes, all 75% yellow and 25% green. Law of segregation. So this is another law. The law of segregation: during the formation of the gametes, eggs or sperm, the two alleles responsible for a trait separate from each other. Now, alleles for a trait are then recombined at fertilization, producing the genotype for the traits of the offspring. Now let's apply the law of segregation. So we did that actually in the Punnett Square, wherein the female parents, the gametes, are segregated on the top part, as you can see here, and then as well as the male parents on the left-hand side. And then by law of segregation, they actually pair up to produce a phenotypic ratio of 3:1. Over here in this example. Now, law of independent assortment. So what is it? Now, alleles for different traits are distributed to sex cells and offspring independently of one another. This law can be illustrated using the Dihybrid Cross. So this is when we're dealing with more than one trait. So what is a Dihybrid Cross? So this is a breeding experiment that tracks the inheritance of two traits. Now, Mendel's law of independent assortment basically explains this, that each pair of alleles segregates independently during the gamete formation. And the formula here is 2 raised to the n, wherein n is the number of heterozygotes. So remember, 2n is heterozygotes. As you can see here, how many gametes will be produced for the following allele arrangement? So let's take a look at these. If I have round and yellow, and then these are just like a bunch of letters that shows you the number of gametes produced for the following allele arrangement. So in this case, there's like 2 different alleles. 1, 2, 3, 4 on the second, and there's 1, 2, 3, 4, 5, 6, 7, 8, 9 alleles. As you can see here, we can deduce that number 1 has 4 gametes total. So that's why there's RY. So you have to combine the R and the Y. Big R, big Y, big R, small y, big Y, and then small r, small y. So that's why you got 4 gametes. Now for this one, there is 8 gametes total. So same thing, you combine all the A's, the B's, the C's, and the D's with different combination, resulting to 8 gametes right here. And last but not least, for number 3, we have your 64 gametes. Of course, I'm not going to put that all together, but if you get the idea of how you can count for how many gametes there are, that would depend on how many traits there are in a cross. But let's go back to the dihybrid cross. So let's say, for example, we want to combine and take a look at the dihybrid cross for seed shape and seed color. So the alleles involved would be a round and wrinkled shape or yellow and green color. Okay, so that gives us the capital R, small r, big Y, small y, crossed with that same dihybrid cross. So this is basically the gametes that we have total. Okay, so that's basically what we have. So in a dihybrid cross, we have this big giant Punnett square over here. And as you can see, we filled it out automatically by putting together and crossing all the gametes at the top, female parent, and the male parent on the left-hand side. And we end up with round yellow, 9:3:3:1 phenotypic ratio right here. And as you can see here, this is basically how it looks like after we converted the dihybrid cross to see the ratio, both allele and both genotype and phenotype of this cross. Now, just to summarize the Mendel's law, let's start first with law of dominance, that the parents cross if the dominant trait is the one that masks the other. Then we can say that if we cross dominant and recessive genotypes, it will result to 100% heterozygous trait or a hybrid trait. Now, under law of segregation, when you cross parents that are heterozygous, then the resulting offspring would be 75% tall and 25% short. And in our independent assortment, this is actually very evident in our dihybrid cross, wherein we have two traits crossed, resulting into our 9:3:3:1 ratio. Now let's move on to incomplete dominance and codominance. So when you say incomplete dominance, F1 hybrids have an appearance somewhat in between the phenotypes of two parental varieties. So for example, Snapdragon's flower, red and white, when you cross, it is not actually one or the other, but instead it results into a different color, which is pink over here. So that is incomplete dominance. Now the second, codominance, where two alleles are expressed or multiple alleles in a heterozygous individual. So an example of this is a blood type. So type A, it can be IA, two capital A's, or IAI. So there's like a dominant recessive, but it's multiple alleles. So these are some examples. I'm not going to go into that, but just to show you how a codominance problem looks like. So let's say, for example, a homozygous male, type B, that's his allele, is crossed with a female. As you can see here, you can say that there's a 50-50% chance of having type A and type B. Here's another example, a type O male, which is two small i's, and a female, which is a B. There's actually a 50% chance of having a type A and type B. Now another is sex-linked traits. Now these are traits where genes located on the sex chromosomes. Now sex chromosomes are X and Y chromosomes. XX genotype for females, XY is for males. And many sex-linked traits are carried on the X chromosome right here. So here's an example, eye color in fruit flies. So the fruit fly eye color is found in the X chromosome. So here's an example. So if it's a red-eyed male, as symbolized here, it's on the X chromosome. The red-eyed gene is on the X chromosome. It's crossed with a white-eyed female, as you can see here. So white eye is a small r. And when you cross them, let's take a look. When you cross them, the Y chromosome in males do not carry the trait. It's just the X chromosome. So the capital letter R is the red eye, and then the capital and small r is still red eye, and then the two small r's is white eye. So when you cross them, this is how it's going to look like. So you can see here, if the female X, two small r's are there, and then the guy carries the red-eyed, you can see here there's 50% chance that there will be a red-eyed female and 50% white-eyed male. Another example of a sex-linked trait is hemophilia. Women are carriers of this genetic trait, and men have the phenotype more often than female. Female are carrier, normally hemophiliacs are normally men. Now here are some examples of more Punnett Square. So if I have a purebred tall and Adora pea plants, it produces an F1 generation, and all are heterozygous. Now if we breed the two heterozygous plant, as you can see here, 25% will be pure, 50% will be heterozygous, and 25% is your recessive trait. So I hope this helps in understanding what our lesson is about on Mendelian genetics and inheritance. And I will see you in class. Bye.